Okay, today we're going to be talking about the kidneys and the urinary system. So, first we're going to go over some of the anatomy of the kidney. You have the kidney, which is here labeled as 1, and that goes through the renal pelvis and then into the ureter, then the bladder, and then the urethra to be peed out. The kidney is fed by the renal artery and vein and goes out through the renal vein and then sends it out through the inferior vena cava and is fed by the abdominal aorta. So today we're going to go over a little bit of the nephron, some of the anatomical features of the nephron I have here, but it starts out with the glomerulus. So there's this thing called the renal corpuscle, which is the glomerulus plus the Bowman's capsule. So your endothelium, glomerulus blood membrane or barrier membrane, and the parietal and visceral, so your podocytes. So this is going to be fed by your afferent arteriole, and all your blood's going to come through here, and about 28% of this blood going by is going to be actually retained by this nephron, and it's all the plasma in your blood. So anything too big for this glomerular basement membrane can't travel through. This is done by the fenestrated slits in there that they only let things about 25 nanometers through, and then there's this thing called the nephrin, which only lets things past that 7 to 9 nanometers through. So anything too big can't go through here. So these are like your red blood cells, your white blood cells, anything large albumin, anything with big charges, they can't go through this. So that leaves the only things that can go through are anything with is either really small and has very little charge. So things that can go through are sodium, chloride, potassium, calcium, magnesium, sodium bicarbonate, glucose, amino acids, lipids, urea, creatinine. Those types of things are going through here, and that is your glomerulus. Now we're gonna zoom out a little bit, and we're gonna go to the proximal convoluted tubule. Proximal convoluted tubule is significant because it does about 70% of the filtration. So it reabsorbs about 70% of the sodium chloride and water, and 100% of your glucose and your amino acids. So it's really doing a lot here. It's like the MVP of the kidney and the nephron. So he's in charge of sending all that out. As you can see in this picture, is a good picture of it. He is really sending all this stuff out right there. So 65% or so, 70% or so of sodium, water, and then pretty much the rest of everything else. Then you're going to go down to the descending your descending tubule. This helps to form what we'll call as the loop of Henle. And going down this descending tubule, your concentration actually is going to get lower and or higher. Your osmolarity is going to get higher and higher, and so you're going to be filtrating out or reabsorbing more and more water. This is due a lot because of the ascending tubule that is pushing out a lot of ions. These ions are making the osmolarity outside of this tubule really high, which is pretty much forcing the water out. So as you head up the ascending tubule, you'll get to the distal convoluted tubule, and there's an early and a late distal convoluted tubule, and this has a lot to do with maintaining your calcium, the rest of your calcium, sodium levels, and maintaining some of the protons. As you get later into the last part, your distal tubule and collecting tubule, that's a lot of where your acid-base regulation, homeostasis, comes from through these intercalated A cells and intercalated B cells. Your intercalated A cells take care of acidosis, and your intercalated B cells take care of alkalosis. So if your pH is really low, protons are high, your intercalated A cell is going to be taking care of this through excreting this bicarbonate, which is then going to take on these protons and get them excreted. This is all done through carbonic anhydrase. That's done in the intercalated B cell as well. But protons are mainly regulated through these two, and this is how you're maintaining your pH as it goes in here to the bloodstream to be reabsorbed. A way that the body also gets rid of these protons is through ammonia, and that nitrogen can pick up another proton to be excreted out. Now, you have these things called principal cells. Principal cells are in charge of mainly your water uptake. So there's this thing called ADH. ADH is activate a G protein, coupled protein that turns on adenyl cyclase, then CAMP, and then PKA, which then goes to aquaporin 2, and that tells all the aquaporin 2s to open and to absorb more water. So ADH is in charge of a lot of your blood volume, because it tells you how much water you need to be reabsorbing in this later part of the kidney. Now, say this weren't to be activated and this water wouldn't be reabsorbed, then it would just keep heading down this collecting tubule and eventually be just urinated out of the body. So aldosterone goes in, and it can go in and activate some cells to, let more sodium and potassium into the bloodstream, also to maintain some more water. So the aldosterone and the ADH, if you remember from our last unit, had a lot to do with the ROS system. And pull up a diagram of the ROS system here. When your kidney lets off this thing called renin, it's going to activate a series of events which eventually leads to angiotensin 2, which leads to aldosterone, which is, sodium and chloride absorption, which tells more water to get in the blood because of an osmolarity thing. And like I said, ADH is also influenced by this angiotensin 2. So now let's get into a little bit of what's called GFR, which is glomerular filtration rate. This is the rate at which kidneys are filtering the blood. This is usually about 125 milliliters per minute, and it is influenced mainly by your NFP, which is your net filtration pressure. Three things. Net filtration pressure is a lot like what we did in the capillaries, and this is usually mainly regulated by the hydrostatic forces in the glomerulus and the Bowman's capsule. The hydrostatic pressure of the glomerulus, that's the main outward pressure. That's pushing a 55 millimeters of mercury, and that is pushing that out. Then you have your hydrostatic pressure, which is 15 in, as well as your B cup, which is your blood colloidal osmotic pressure, which is doing about 30 millimeters of mercury in, and that's created by your blood proteins, your albumin specifically. So your NFP is usually about 10 millimeters of mercury, and this GFR, which is the rate at which your kidneys can filter blood, it's regulated in two ways: through tubular, glomerular feedback and myogenic feedback. Tubular glomerular feedback occurs in the macular densa, which is in between your ascending limb and your distal tubule. Your lowering blood pressure would tell the macular densa to regulate NaCl levels, and if your NaCl levels are too low, that's going to tell nitrogen oxide to vasodilate and increase blood flow to get your blood pressure back up. And high sodium chloride, the exact opposite, the vice versa. So your macular densa and your afferent arteriole make up the juxtamedular apparatus. Now, myogenic autoregulation, that's your intrinsic smooth muscle. And so that higher blood pressure through stretch-activated calcium, which vasoconstricts and reduces blood flow, and glomerular pressure goes to normal. So, yeah, that is some of the kidney.